Miramontes. I'm very happy to be here and having the opportunity to speak in front of everybody. I'm a little intimidated because everybody's research is amazing, but just to give you a little bit of background about me, I started working with Danny Yoshizima through a URAP program last year, so it's been about exactly 1 year since I last presented. I started working with medical imaging, and then eventually I transitioned into working with fibers for quality assurance. So thank you, Stefan and Danny, and everybody else for putting this together. I really appreciate it. I've learned a lot, and I've enjoyed watching everybody's presentations. Also, I want to thank again Danny for as a collaborator and doula up at the ALS, which is where we got our data from. And yes, okay. So I will be talking to you about convolutional neural nets for fiber detection models. So what fibers am I talking about? So the material we studied are called ceramic matrix composites, which you can see on your left. They're 3D rendered. That's how you analyze matrix cracks. And then on your right, you can see that that's how they look like when you look at them up close. So they're up close. So they are like tube-looking things. So there is an increasing demand for these materials because they're lighter and that they withstand higher temperatures and they are damage tolerant. So these are particularly used in jet turbines and jet engines, and as seen in the image below, GE has been developing the next generation CMC components just to build turbine blades. The quality of these materials, which is where this whole research is based off, depends on stress test imaged using microtomography, which allows us to study the fiber distributions seen in a fiber bed, which I will show you images of this so you know what I'm talking about if you're not familiar with this, fiber pullouts or matrix cracks. So besides quality assurance, what other motivation is there to study materials that look like this? So there's a growing frequency in which micro-CT data is acquired. So with this growing frequency, there's an ongoing need for the transformation of these experiments into scientific discovery. And this scientific discovery demands new analytical tasks with higher accuracy and scalability. So my favorite question as an aspiring data scientist is, what can we extract from micro-CT data to examine the quality of CMCs? Or can we study the current models to discover new characteristics of the specimen? So to address both of these questions, actually they're numbered. So the first one is our preliminary studies, and the second one is where we're at right now. So the data that we utilize is publicly available at the Material Data Facility, which is based off the work of Natalie Larsen and Frank Zock. The number one around the square, that's the first data set we use for our preliminary approaches, and then the last one on the further right is the one we use for our ComNets. So to study micro-CT data, ideally we would go through all this pipeline, right? But things don't always go as expected as scientists. We all know that. So we have the micro-CT at the beginning, which is where we collect data. Then we pre-process the tomographic reconstruction. We filter and segmentate data. We do the information processes. We extract this, and then we visualize this information. Then we do the virtual specimen generation. Then we do the algorithm development simulations, and then we compare it with time-lapse data, and then we understand and predict, and we tell GE what they need to do about their materials. But getting there is a little bit more complicated. Right now, where we're at right now, the first 3 steps are already assumed to be done for us, and what we focus on right now is the information processing and the information extraction and visualization. So here are some of our preliminary approaches, which were done in scikit-image. So the very first one we did, which should be familiar if you were here in the tutorials on Tuesday, Danny and Alex went over some of these. So these I did by following the tutorial, so if you're interested in this, I suggest you go on their website and give it a shot. The data is publicly available, so have at it. So the very first row shows blob detection, which you can see at the top, top left, there is some good detection of the fibers, but there's a few false positives there. Then we also attempted to crop one of the image slices available from the 3D specimen and said, okay, let's just try to calculate this area and use a Canny filter and then see if we can count these to see if we can therefore detect them. That did not go as planned because some of these fibers are agglomerated together, so it's really hard to we can't pull them apart, but it's hard for the computer to see, to distinguish one fiber from the other when they're so close together. We also tried half transforms, and we weren't that successful. Not to abandon ship completely a scikit image, but we said, let's put this aside, and then we said, okay, if we start studying some of the other data sets available, which is the one on the further left I was talking about earlier, when we started looking at the segmentations that were already done by this research group, we started looking at some abnormalities that are seen on the segmented image, on the binary image on your on the bottom right. And we said, are these that accurate? So we said, okay, you know what, let's start playing with these. What if we take the center of this specimen and we actually manually label all of these fibers, train a neural network with our manual labeling, and then test the remaining data with the neural network. So that's what we're at. So here's an image of my pipeline, or the pipeline that we're working on. So we have the CMC specimen over there, and then I'm going to talk about how we how we extract the CNN inputs. So first, I manually labeled each fiber, a little painful, but it has to be done. There were about 5,000 clicks. And from there, I used the already segmented results to create another data set. So what did this yield? So this led to the creation of two data sets. The baseline data set is based off of my clicks, and the other data set is based off of the already segmented results that Natalie and her team had. So again, the baseline data set is used for the training of the ComNet, and then the segmented crops data set is used for the testing of it. Another approach we took is we said, okay, let's do some max projection and see what we can learn. So we took the raw image, and then we calculated the max projection, and then we arrived at a lossless data reduction because within this micro CT image, there is so much of what you don't need. You want to get essentially to the fibers, and you want to avoid the areas of the fiber of the specimen that don't contain any fibers. As seen at on the image, the very first one I showed, the CMC matrix is voids or where there is no fiber, and the silicon carbide fibers are the ones we're interested in. So a little bit more detail on this data generation process. So the inputs for the CNN created the baseline data set, which created about 8,000 images, 10 by 10 each. And then the segmented crops generated that many images. And you could see that these actually take a long time to generate. So with all those images to test the neural network in, that's also going to take a lot of time. So we did a few testing on a slice, and we saw that it was going to take quite some time. So we only tested this on 10% of the data. So we did every 10th slice. And the reason why we chose this is because there is less likelihood that the material changes in every 10th slice. So we said, look, let's just use the crops we generated to test this. So what are the outcomes? So before I get to the CNN outcomes, we also did some statistical analysis, and we found that when studying the segmentations already done in the second data set, we saw that there was a large variation of fiber cross-sectional area, which was beyond the reported 13 to 20 pixels in the semi-automated model I'm talking about. Additionally, the biggest one, I believe, is the fiber counts across the stack indicate errors in the segmentation model, particularly on the high, lows, and peaks. Like, I don't have a pointer, but you can see that it's not a good sign that there's such variation on fiber counts. And here are some of the results for the CNN outcome in terms of accuracy, precision, and sensitivity. So what does this tell us? What did we do? So We were able to achieve a qualitative and quantitative analysis of fiber models using CMC that have been used in CMC manufacturing. So in conclusion, we were able to emphasize a large variation of fiber cross-sectional area going beyond the expected diameters specified by the manufacturers. We also found fiber count oscillations indicating, as indicated in the previous slides, since the fiber breaks were not expected in the CMC experiment. What's a fiber break? Well, if you look at the specimen and there's, let's picture a tube, and in some image slice of that fiber you were looking at is not there anymore in a particular slice, and we consider that a fiber pool. But that was not expected in these experiments, so this is what we're talking about. Additionally, the CNN we trained and tested might serve as a method to probe the segmentations made by other scientists, which will in turn help us eliminate false negatives from models that contribute to the lack of fiber continuity, the fiber pool as I was talking about, within the micro CT stack. So what's next? We want to account for the wider variability of fibers, not necessarily present in this data set, but because I've worked with other data sets, I know there's like variation on the sizes or the fiber coding of that, which would be like the edges of the fiber itself, and that can definitely alter your algorithms. We also want to explore the remaining image stacks. The image stack we were working on right now was just one of, I believe, the 4 available. And then we also would want to try different CNN architectures, as well as other augmentation protocols to increase the sample data. This will lead to the increase in robustness to fibers, to the fibers of varying size and different deformations in our model. And that's it. I will be happy to take any questions. Also, if you want to look at the classification portion of this, that's available at that link, where you can see my whole journey of creating the data set and feeding it into the already trained network. Thank you.
We have time for 1 or 2 questions for Silvia. Mike's live.
Well, first, Silvia, I'm really proud of you.
Oh,
I mean, you started this work last summer as a part-time. You were working for free for quite a while, and then while you were attending classes from Fernando Perez, the guy that worked on the Jupyter Notebook and invent IPython. I know it was really tough, and you've been taking on this project since then and going through all of that. So this is really exciting. How about you tell me a little bit of how these 2 portions, I mean, when you showed the picture of the CNN, like the big diagram.
This one?
Apparently there are 2 things happening here. Can you elaborate a little more?
Yes, I can. So I should have labeled these. Oh my God. Okay, let me just load all of these. So I would want to call the bottom the segmentation at the fiber level. So to really get in the nitty-gritty and kind of ask, like, did you get all the fibers you should have gotten in your specimen? And then I'd like to say that the bottom, the max projection approach refers to the segmentation at the image cross-section level, which in turn leads into lossless of data reduction. So you don't want to be looking at things that you don't need to be looking at, if that makes sense.
But it's also the case that the top one, it's really independent on any sort of segmentation, and the bottom is more towards, well, you have some sort of segmentation of how good it is based on these standards that were published in 3 different papers by Natalie, right?
Yeah.
Okay. Thank you.
Cool.
Any other questions for Silvia? This one over here.
Thank you for the very nice presentation. I think it was really cool that you showed the data as provided. I know Danny and you folks are really leading the way for the synchrotron community that is very, doesn't have a tradition as a lot of the other communities of putting data out there. You talked about the materials data facility. What do you think is the best practice for people going forward to publish their data? Is that the best practice?
In terms of
If they want to
citing-
publish their data, like this.
like publish the data sets that like
Yes.
um
How should they do that? If they're working in this domain, what is the best practice?
well, I particularly don't have any experience in publishing large data sets, but I can tell you that I benefit from people that actually publish it in open source data because it allows me to experiment and do all kinds of crazy approaches. And it allows me to switch paths if I know that one data set did not work. I'm going to say, okay, let me try another method and another one. Maybe that will stick and that will work. So I don't know if Danny has another input in terms of...
I have an answer to that question, I think. So
Okay, great.
at least because so there's been yearly kind of a meetup between the cyclotron community, and I know that there's a movement to establish Tomo Bank, which is a like collection of available data sets. They have like some data from like published papers, and then also have some like round robin sample that's been flying across the world and gone to each synchrotron. So there is this kind of, it's building up. I don't know the exact status, but they are building it, and it's online. I don't know how many people are using it, but there's a fair amount of data there.
Yeah, so to try to answer it, I would like to take on the whole credit, that some of the material that Natalie published was because of me pushing her and bugging her, but I will actually give credit to ImageXD. She was here last year, and we all were bugging her and telling her the importance of sharing her data set, and that became, really important both for her publications as well for everybody else that is trying to improve on the method that she built. So I would really encourage you to find ways to share with us or through some other, it can be through materials facility or Tomo Bank or whatever other ways you benefit the community. You have more references to your work as well as brilliant students like Silvia Miramontes to really progress on her studies.
So
Any other questions? Dula?
Okay, so I think let's thank our speaker, Silvia.
Thank you.
You had him? Is here for you.
All right. Thank you.
So, come on, technology.
Nice.
Beautiful. Okay.
Okay. I'm actually going to change the display really
Okay.
quick.
That's fine. So our last talk of the morning